Okay, so real quickly, so Stan, this is Ann again. Thank you for having us. It's wonderful we get a chance to share some of the resources that we have down here in San Antonio with all of you. Um, and I'm going to do about 10 minutes of presentation here. I'm going to give you an overview of the CIDD since it started, and a little bit on our medicinal chemistry and synthesis core, which is one of the two core facilities. And then I'm going to turn it over to my colleague, Matt Hart, who's going to go into depth on the high throughput screening core. What is the CIDD? Where did we start? So we're in our 8th year. We were founded in 2012. And the whole idea behind the CIDD, at least within the area of San Antonio, was that there was a real unmet need for translational research for small molecules. And I think that's true everywhere, and I think we all know this. And so the idea behind our mission was to provide two key resources that you don't normally have in an academic setting, and use that to develop small molecules, both the high throughput screening core to identify leads, and a medicinal chemistry core to optimize those leads. And we work with both industry and academic collaborators, and I'll highlight some of those. And so we have a pretty organized structure. I want to just take you through our structure real quickly. So Matt and I oversee the operations in the two core facilities. Myself and Bruce Nicholson are the CIDD directors. We have a steering committee that we meet with frequently that consists of sort of local leaders in San Antonio. Obviously, we have UTSA and Health Science Center leadership that are supporting the CIDD. Dean Silva and Audrey Lamb on the UTSA side, and Rob Romas on the UT Health SA side. And then we have two advisory boards. We have an external advisory board that is made up of John Lowe and Craig Dion, which both bring an enormous amount of pharmaceutical experience. And we also have an academic member as well with Brian Blagg. And then we have an internal advisory board with some of the leaders, again, that are in the San Antonio area, namely both in research as well as in our tech transfer and commercializations offices. We're associated with a couple of different cores that we work closely with. Our other structure-based core facilities, NMR, X-ray, etc. And then Matt might mention a little bit about the siRNA screening core facility as well. So I'm going to take you through some of the details of what type of projects we work, what our resources are, our capabilities, and sort of where we work. I think this slide's probably a no-brainer for everybody. You can see that for the most part, CIDD works from early target discovery with collaborators all the way to preclinical studies. And the majority of our time is obviously in lead discovery medicinal chemistry. But as you're going to see with one of the examples I give, we do it, at least in the medchem side, we venture into development quite a bit in the form of formulation development, salt screening, process improvement, and synthesis, etc. Here's just a snapshot of some of our collaborators we've been working with over the years. So both in academic and private industry, we do a lot of work with private industry as well. The ones in red are ones that actually have active projects ongoing with us right now. But you can see we work with a lot of the people here in Texas, outside of Texas, and we also work with some of the private companies, both in and outside of Texas as well. So here's a little snapshot of what our CIDD medicinal chemistry core facility looks like. So it really is a state-of-the-art facility. It was a brand new lab when I established it in 2012, and we built it to be more like a pharmaceutical research lab, like you'd find at a Merck, at a Pfizer, or Lilly. And so we really have some pretty nice automated equipment. We do a lot of parallel synthesis with block reactors, a lot of automated purification. We have extensive analytical capabilities, so 2 HPLC mass specs, prep HPLC, analytical HPLC. And the beauty about the CIDD that I really like and is I think helped with our success is all of these resources are dedicated to the core facility. These are not shared with other members of the department. And so the work that we do medicinal chemistry synthesis in the small milligram quantities to gram scale. We can do a multi-step synthesis. We've done natural product total synthesis up to 26 steps. We can do shorter syntheses as well. A lot of our programs are structure-based drug design programs. We also do a lot of probe molecules, biotinylation, tagged molecules. We help people with that as well. And as well as I mentioned, natural products, synthesis, and analogs. One of the things we've been building over the last year, I'm going to talk about in a little on the next slide, a little more detail, is a ProTac platform, and I'll mention that in a little more detail. So since we've started, we've worked with a number of different collaborators across Texas and across the nation. We probably worked on, I think we're up to 74 programs now. Just the MedChem side, we on the UTSA side, we've brought in a little over 17 million in extramural funding. Matt's got some other statistics from the other from our screening core facility as well. One of the metrics that we've seen, I'm sure other core facilities have seen this as well, is we've seen a real increase in the success rate of grants that incorporate these type of technologies. And so we've seen that here locally in San Antonio as well. I'm not going to go through our portfolio, but we're about, if you look at all of our programs that are active right now, we're about 60% cancer. We have about 10% in CNS and pain, and then another 10 or 20% in infectious disease and diabetes. So that's a large majority of them. I think Matt has another breakdown as well for what's going on in the screening facility. And these are just some pictures of some of our laboratory capabilities and resources. So let me just finish up with, before I turn it over to Matt, a couple of highlights of programs we've done that'll sort of give you a flavor of the type of work that we do. So one of the programs that we've been working on now for about 2 or 3 years is with our collaborators up in Austin, PKC epsilon inhibitors. These are for non-analgesic pain indications. It's a multi-site collaboration with UC San Francisco, Austin, and San Antonio. And this is a great program where we've designed and synthesized probably over 100 different compounds across a couple different chemical series. We've got X-ray structure. We're developing SAR for selectivity, brain penetration, and our lead compounds are now showing efficacy in vivo models. So it's a really exciting time for that program. And that's funded through a DoD grant with Bob Messing in Austin. A non-cancer program that's received a lot of attention here that we've been working on. We're in our 6th year on this program as a large program developing novel anti-schistosomal agents to combat some of the resistance and sort of multi-species activity issues that's with the current therapies. We've developed, again, using structure-based drug design, looking at X-ray, we've developed some really novel chemical matter that are not only active against all 3 species of schistosome, but are extremely potent as well. And this program is geared to now where we're starting to look at in vivo efficacy PK for our lead molecules. Those are both academic collaborations. I want to give you a flavor for what we do with some industry collaborations. Not showing any structures, but we worked with a private cancer company that was actually a separate funded cancer company, and we did all of their medicinal chemistry work for them. We synthesized over 500 analogs across 3 chemical series. This was over the course of about 1.5 years. We optimized SAR and physical chemical properties, and then as they selected lead molecules for their in vivo screening, we went ahead and we did large-scale synthesis to support those materials. We also did salt screens for aqueous solubilities and drug stability properties. And then we did a full tech transfer package to cGMP. So that's about the cutoff for us in the MedChem. We obviously can't do cGMP synthesis, but we've done process optimization where you could take that synthesis route and hand it over to a cGMP manufacturer. And then finally, it's sort of a platform technology that we're working on that we've been building is the proteolysis targeting chimeras, or ProTacs. Obviously, these have mostly been targeted for cancer, but other indications are looking at these now. And what our platform exists of is purely right now on a chemistry basis. So if you look at the ProTacs that are out there, usually the targeting element and the ligase recruitment element are defined. And what you put in between those two in the form of a linker really dictates a lot of the drug properties. And there's several publications of this from both pharma and academic now showing this. And so what we wanted to do was create a platform of linkers that change physical chemical properties, rigidity in terms of rotatable bonds, and also be flexible with the types of chemistry you can use to employ these. And so anybody that wants to work with us on these PROTAC compounds, we sort of have a library of already assembled of really unique chemistry for putting these together, and unique structures for this element of rigidity. So this is something we're going to continue to grow as well. So for anybody that's interested in PROTAC type programs, please come and talk to us. With that, I'm going to just, before I hand it over to Matt, I'm going to tell you, this is where our funding looks like and our collaborations. We have NIH, CIPRIT, DoD funding, and we also get a lot of philanthropic support here as well. The Max and Mimi Tomerlin Volker Fund have been very, very gracious to us, myself and Doug France. And we've also just recently developed funding through the San Antonio Partnership for Precision Therapeutics as well, which is another group that's growing here in San Antonio. And I'm going to leave you with this: our full-time staff in the CIDD MedChem facility are really top notch. Leo Wang, postdoc out of UT Southwestern, has an extensive amount of synthesis experience, as does Karenelle and Marie, our 3 PhD synthetic chemists. Mike Tidwell is a master's level, but comes with 7 years of experience from Eli Lilly Neuroscience. And Bismarck Campos is our bachelor level chemist that works and has over 16 years experience in working in industry. And so I would put this synthesis team up against anybody, so to speak. They do very well, and we love collaborating with people. So with that, I'm gonna turn it over to Matt.
Okay, thanks, Dan. So let's see, so I'm gonna share my screen. Okay, so I'm just gonna jump on to the next slide. I don't know why that's not working. Okay, so I'm not gonna spend too much time on this. So basically the area that is covered at the high throughput screening facility for CIDD encompasses a lot of assay development. So we work extensively with people. They come to us and kind of talk to us about their biology and how that can be kind of converted into a high throughput screen. I spent a lot of time working with people on this, and then optimizing for HTS, and then running through screen hit picking, determining potency and selectivity, and then moving those projects on to MedChem, where you kind of develop this kind of loop before things move on to preclinical. Also introduced here is the target identification. So that's a capability that I'll get to later in the talk. So this kind of provides some information about the therapeutic areas that are covered. Again, as Stan said, most of it's represented in the area of cancer, but we do have other areas that we do help with. We're seeing more kind of infectious disease targets or projects coming up, especially with the COVID-19 issue. So, and aging is also represented here. There's a lot of excellent aging researchers at the UT Health coming out of the Barshop Institute. So again, we've worked with people over the last 8 years on 115 different research projects that's numbered 84 investigators. And I've been involved in 150 grant submissions. 76 of those have been funded. We have 22 grants pending. And when you incorporate program projects, NCI funded program project, as well as program projects through NCATS, we've brought into the university about $100 million worth of grant funding. These funding sources are program projects, U01s, P30s. We have a P20 out, NIH R01, R21, CPRIT, DoD, and a number of private and internal pilot award mechanisms. So the personnel, again, I come in with over 20 years target discovery and drug discovery research. I've got 2 full-time PhD level scientists, 1 research scientist who puts a lot of his time into doing protein production. So we also have the elements of doing protein production in SF9 cells, as well as E. coli. And that's been helpful for some of our, for quite a few of our projects, and as well as a research associate. I'm also looking to hire a data analyst person. So if there's someone out there who's listening who would be interested in moving to San Antonio, please give me a shout. Some of our core capabilities, again, we have a chemical library that numbers about 170,000 compounds. We do screening of purified component cell-based assays. We've got plate reader-based assays, as well as high content screening using the PerkinElmer operetta. In the RNAi facility, we have the Vol of Sciences IC200 imaging system. We have liquid handling capability, including the echoacoustic dispenser, and some of our data analysis tools include the Columbus image analysis, as well as the Cambridge or the TIBCO Spotfire analytics program. And again, a lot of these services really, revolve around determining the drugability of a target and screening design. So some of the libraries that we have, the first three, those are all kind of FDA approved or well-characterized bioactive compounds. And so we have that from three libraries. One is a low-pack, Prestwick FDA, and then a Live Chemical Bioactives, which is pretty well annotated. It's 8,000 compounds, targets a variety of different known targets, numbering probably in the neighborhood of 600 different targets. For diversity, we've got a Mainbridge hit finder, but we've both been, more focused recently on these live chemical libraries. One is a FSP3, which has greater kind of three-dimensionality, and their live chemical diversity set. We have a UTSA select library. These are unique compounds that really have been sourced at a UTSA. And recently we've added a covalent inhibitor library, an ion channel inhibitor library, and now we're sourcing for more complex macrocyclic compounds that'll occupy a unique space. And these are going to be designed for, applying to targeting protein-protein interfaces. These are just some of the projects, or at least some of the readouts that we utilize. TR-FRET, fluorescence intensity, absorbance for, we've targeted two different phosphatases, high content imaging, looking at cell-cell coupling or mechanisms, small molecules that can promote PD-L1 degradation. We do quite a bit of fluorescence polarization type assays using for DNA repair enzymes or targeting RNA binding proteins such as Musashi or ALKBH5. And again, high content imaging targeting signaling pathways, nutrient activation of mTOR or mitochondrial calcium flux. So we've more recently, as Stan alluded to, with the development of PROTACs, really trying to get into this undruggable target space. One of those, and so in order to do that, we've kind of made purchases of covalent inhibitor library. We've used that on about 4 different targets right now. And I'm quite happy with that because we don't see a high hit rate. So, I'm thinking that, with this, even though they're covalent, we could still get some specificity and then further optimize that with MedChem. We're also, going to invest in this macrocyclic library. And I think that, again, as I stated, these will be, potentially or useful in inhibiting protein-protein interactions. So this next area that Stan talked to is getting into these PROTACs. And so the method that we use for trying to identify small molecules that bind to a target or bind to an undruggable target, or previously considered undruggable, the approach that we're going to use is microscale thermophoresis. And basically what that involves is that it's basically studying how a protein migrates in a thermal gradient. And so, you can bind, and look at complex formation or even binding of a small molecule to that target. And you can see how it changes, the thermophoretic ability of that protein changes when a small molecule is bound. And so we have an instrument, it's fairly high throughput. It's from a company called Nanotemper. So far, we've used this approach against 3 different targets. And one is EZH2, which is an epigenetic modifier. We're starting work looking at proteins involved in COVID-19, as well as DNA damage targets. And again, like the covalent library, when we use just our diversity library, we don't see a lot of, the hit rates are really quite reasonable. So we're getting hit rates initially in the area of from 0.3 to 0.5%. And then, we can do further kind of triaging based on dose responses to these that we can determine in this approach. The other aspects that we're adding, we're going to invest in a SBR instrument from a company called Nicoya. That's nice because it's really an easy to use SPR instrument. And we're also going to try and get thermal stability assays up and running. I think that those 3 different approaches really kind of complement each other. And the other thing that I bring up is the RNAi high throughput screening facility. So again, with that, we have RNAi screening, but we also have at the GCCRI really good statistics and bioinformatics pathway analysis capability. And the idea is to identify druggable targets or even non-druggable targets and then feed those into our pipeline in the small molecule facility. So we could run high throughput screens, or we can also run screens on different subsets. We've been doing a lot of kinome synthetic lethality screens with a group at MD Anderson focused on breast cancer cell lines and looking for ways of enhancing response to eribulin or TK inhibitors. We've also done whole genome synthetic lethality screens with antibody ADC conjugates. And then we're also at the RNAi facility, we also have CRISPR screening capability. And so we've done a few whole genome synthetic lethality screens with that approach. Again, so at CIDD, obviously MedChem and small molecule discovery capabilities are available. Stan alluded to X-ray crystallography. So I also want to point out to people that at the Health Science Center, we have X-ray and NMR facilities. And we recently hired Sean Olson. He's going to set up for cryo-EM. So there's other kind of structural capabilities at the Health Science Center for people to take advantage of. And so this kind of integration, we really want to try and have the small molecule facility and the RNAi CRISPR facilities really kind of complement each other and drive integration. And I just want to thank the people that have worked with me at the 2 facilities. And here are some examples of funding that we've acquired over the years. All right. So, thank you.
Thank you, Matt, and thank you, Stan. Very much appreciated. We'll hold questions till the very end. So right now, we're going to turn it over to Cliff Stevens. So Cliff's academic and research interests, they're focused on drug discovery, both new chemical entities and repurposing of existing drugs. Cliff was formerly the director of high throughput screening and new leads discovery at Incisive Pharmaceuticals here in Houston. So he brings that biotech experience into the academic side. He transitioned from Incisive over to initially UT Health Science Center back between 2007, 2012, and then recruited over to IBT, the Institute of Biosciences and Technology at Texas A&M. So currently he leads the CPRIT-funded GCC combinatorial drug discovery program at IBT, and I believe this is now into the 2nd, if not the 3rd iteration of funding for that resource. And so we're delighted to have Cliff give us some background on this great resource that he runs. So back to you, Cliff.
Thanks, Dan. Appreciate the interaction. Thanks, everybody, for coming and joining with the meeting today. I'm going to just run through some basic background about our core facility. What I'd like to emphasize, as you might see as we go through this, is the flexibility and versatility of the core, and we've tried to build that into the program, and I think you'll see that as I go through some of these slides. Our bulk of our funding, or all of our funding primarily, is from CPRIT. That funds about 75% of our program. The other 25% allows us to work on projects regardless of topic or of area of interest. So we are capable of working in not only in cancer, but also in areas beyond that as well, and have done so with investigators throughout the Houston and Galveston area. As Stan pointed out, we're part of a GCC group of CPRIT funded cores. This is just sort of the list. This is on the GCC website. You can access all the home pages of these various core facilities from this website. And you can see that a lot of them are focused on hit discovery. We have a little bit in target validation, and then therapeutic monoclonal antibodies, PK, PD, formulation, and now Stan and his group there at doing the accelerator for cancer therapeutics. So, you know, a lot of tools involved are available for people that are doing drug discovery in and around the Texas Medical Center, Houston, Galveston area. Sometimes people don't know where we are. The building on the left is the Institute for Biosciences and Technology. We're on the 9th floor of that building. Where are we in relationship to the Texas Medical Center? You can see that we're on the edge of the medical center, but we're within, you know, 10, 15 minutes walking distance from just about any institution in the Texas Medical Center, and only about 35 or 40 miles from Galveston. So we have a lot of interactions with UTMB as well. The types of goals of the projects we work on, a lot of times, we have done some target identification, but primarily we do new molecular probes. We do some of that. We're not doing much small molecules. You'll see we're primarily focused on repurposing drugs and also mechanistic screens for mechanism of action, target identification, validation. But for small molecule screens, we always defer to the folks like Kevin Dalby and Stan and their fine groups, and we try. That's not our expertise. We don't do that, but we'll refer you happily to those investigators. We do a lot of mechanistic studies focused on therapeutic vulnerabilities and identification of drugs alone or in combination, with repurposing with the idea of rapid translation to preclinical and clinical testing. We do both small projects and large scale projects. We can do proof of principle and preliminary data for grant support. If you've got a very simple endpoint, those screens can go fairly quick and have a rapid turnaround. But we've also had some projects that have run for years, actually, with large panels of screens, 60 or more cell lines. It's almost like a CCLE type of approach. And we've also ventured into more complex models, which I'll show towards the end. We've done a little bit with organisms, and primarily zebrafish. And we do a lot of live cell imaging and microscopy these days. Some of the targets that we've worked with, again, cell model systems, engineered cells that have reporter constructs, phenotypic screens, and you can see the list there, running from monolayer and suspension cell cultures to tissue chips and organisms. Again, we're a high throughput screening core facility, and this sort of shows the evolution of the core over the years since about 2006 to present, starting off with instruments that can dispense drugs by tip with the TCAN and the Beckman, and then we added pin tools later on, and now as San Antonio does, we use acoustic transfer for the bulk of our transfers. Output devices, we can do just about all of the standard biochemical assays with 2 automated self-plate feeding, multi-reader with all the capabilities listed there. But the bulk of our projects, being cancer related, tend to be more cell-based type models. And so we have a large focus on confocal, automated confocal microscopy, both for fixed cell and live cell imaging. And we have a number of platforms for those with high throughput capabilities. All of our platforms are automated so that we can get the throughput that we need to do the screening that we're working on. One of our newest platforms that's just coming online right now, which was a an area we didn't explore very well was suspension cells. And so we're with secret funding to start a high throughput flow core that we received last year. This is the platform that's coming online now, which is built around the Bio-Rad ZE5. You can see 5 different lasers and 30 detectors. But some of the unique features of this is it can do small particle detection, so it's good for exosomes. Less than 300 microns. If it's unlabeled, they claim, we haven't tried it, down to 50 if you have a labeled particle. It's good for both 96 and 384. You can see the rest of the features there. Also on the deck, multiple dispensers, washers, centrifuges. So the whole deck will be automated for flow cytometry. Some of the topics that we're doing that allowed us to move in this direction are more like the complex co-cultures and introducing immune components. These were areas we were struggling with and weren't able to do very well. So that we're hoping that the flow cytometry core will actually allow us to do that in a much better way. But we are a combinatorial drug discovery program. Repurposing is the focus. So our libraries are all listed on our website. But you'll notice that they're all targeted agents. So for repurposing, we look at FDA approved drugs, investigational agents. For the mechanistic, we look at drugs or compounds that have known, that were developed against known targets. We can work in 2 and 3D, mostly cell-based systems, both by imaging and biochemical. And most of our combination screens, investigators are interested in either additive or synergistic efficacy of their molecules. We have seen a growing increase in the use of 3D model systems, and it's something that that's one of the reasons why we added the live cell capability. That ability to do temporal studies is really important in these types of models. It's an example of a spheroid that can form spontaneously in the bottom of an ultra-low attachment plate. We're also working in organoid cultures where you can plate single cells in a matrix and then over time see the formation of these organoids. And then we've been working with momentous plates and other microphysiological systems through an NCATS funded program to try to bring some more human on a chip tumor efficacy type studies. And that's an area that we're going to be exploring more in the future. Along with the 3D models came the challenge of how do you analyze the data for those. We have a very good imaging analysis and informatics team, and they've been developing AI-based models using deep neural nets and machine learning and transfer learning to try to take these pictures. Obviously, you can see the complexity of these types of images. It's not just a segment and get feature space on these. They're much more involved with that. And it's required a lot more work. We validate them against biochemical assays, and in this case, we used cell titer glow as a standard for assay viability, and then taking by comparison different modeling ways to do modeling of the data using nothing more than transmitted light images and then the machine learning algorithms. Those can then be translated into efficacy studies where we can actually take the data, do the imaging, and then you can see this one ran over the course of 6 days. We're able to see cell growth, and then we were looking for drugs that would reduce the cell viability and growth in these assays. The next step then was to automate all of this, and so that's what we've been able to do. We've been working on both organoid and spheroid assays, where we can take these and then start to do the screening, both in single agent and combination, using a combination of imaging endpoints for daily monitoring and then a terminal endpoint, so we actually can get a multiplex of data points for viability as well as cell growth over time. This is an example of some of the images that we've been working with. These would be an on-plate controls for one of the models we work with. And you can see the complexity of the model and the negative control. We're looking for a change in phenotype with the positives, and then if the cells are dead, you just get a matrix background. Again, you can apply the image analysis and processing to that. You can use half of the controls to actually train the model, use the other half as a withheld test set and to test the functionality of the model. And we're also finding that we're able to transfer these models. So there is some transfer between different models, different model systems with the model. And again, in this case, we were looking for the probability of these organoid organoid projects, being either did it look like a positive, did it look like the negative, or did it seem to have a cytotoxic response? And you can see that with different drugs, we can see different responses with that. Informatics has been another area that we've been really working hard to develop in the project, and that's where we're trying to add context to the screening results that we do. So we're more than able to do the high throughput screening and collect the data, but it's what happens with the data thereafter. And we've got an informatics group that we've brought on board, and that you can see that they're more than, the more that information they can get, the more that they can incorporate that into the data. You can see just something simple. A simple representation would be drug responsiveness by class, and what we call a tree view, basically. And if you can imagine taking these amongst different cell types or different model systems, and you can actually make comparisons just by looking at this simple tree. You can see changes in size of the circle, which would be the efficacy, and what's coming up. But if you also have genomic data, then we can also start to explore the pharmacogenomic analysis of the data as well, and then put it into the context of that. And so those are areas that we've been really working hard on over the last couple of years, and it's starting to pay some dividends for us. As Stan mentioned, we're fortunate enough to get new funding from CPRIT to apply to the core. And with that, we brought some new research initiatives. And in this case, we're looking at energy metabolism in cells in cancer. And this is an area that we're going to be exploring. It's just starting. It'll take us a couple of months to get things up and started. But this is sort of the idea behind it, is, profiling cancer cells to see, profiling them to see what energy they're using. You can use these profiles as a response treatment. And some of the ways that you can do that, and what I wanted to do with this slide is say what we're not. What we're not is a metabolomics group. We are focused on energy metabolism use in cells or by cells. And the approaches that we're going to take are plate-based. So you can see we'll have access and use of seahorse. And then we'll also be using some imaging, which I'll cover in the next couple of slides, as orthogonal and validation screens using a multi-photon optical system. But if you do need metabolomics, it is available in the Texas Medical Center. And that's a group run out of MD Anderson by Phil Lorenzi. So I thought I'd just give him a plug. But it's a very good group that if you're really interested in looking at metabolites and metabolomics, that's a group that you would want to talk with there. But getting back to ours, our optical metabolic imaging is a collaboration with Alex Walsh, who's a new assistant professor in the Department of Biomedical Engineering at Texas A&M in College Station. She trained with Melissa Scala at Vanderbilt, and her background has been in cellular metabolic imaging and to predict drug responsiveness. And that's what we're going to be trying to leverage as we move forward with this new CPRIT funding. So her work is focused on tumor metabolism as a marker of drug response. There are it leverages the autofluorescence of the cofactors NAD and FAD that are naturally occurring in the cells. And so it's a nice label-free system that's an optical system that you can use. You can monitor as frequently as you want. It's non-destructive, and it gives you the ability to do single cell analysis. You can see here a list of some of the parameters that can be obtained from the data. You can even look for subpopulations of cells within the data. It's been applied in her case to tumor organoids. So we're also going to be trying to apply it to our 3D model systems to see if how those work in the context of drug screening. And you can see just from this simple slide, through a comparison of different types of breast cancers, TNBC with ER positive, HER positive, you can see the differences. Okay.
Sorry to interrupt. I'm not sure the slides are advancing on the display.
Oh, okay. That's not good. Okay. Are you not seeing it change at all? It should be just the right panel. Okay. Basically, but in the end, we're going to automate this platform as well. So it's a multi-photon microscope that we're going to automate with an incubator so that we can do serial. How about now? Is it now changing? Anybody?
No, it's not changing. You
No.
might want
No.
Let me stop and restart them real
to
quick.
stop and restart.
See if that makes a difference. Any better? Should be back to one of the first slides I showed.
Give it a moment. It's still thinking about it.
All right. Thank you.
Doesn't appear to be thinking very positively. It's still.
Well, I'm really near my last, I'm near my last slide. So let me try stop and start one more time. If that doesn't work, I'll just do a verbal at the end. Anything there?
No,
No.
nothing.
No.
Time for a word picture.
Yeah, I guess so. Basically what it was is what's next steps for us. And one of the areas that we're going to be trying to go into in the future is developing, taking advantage of the 3D models and the organs on, or tissue on a chip, and applying that type of technology into fill. As I showed earlier, there's a heavy focus on hit discovery, and there's some in preclinical, but in the lead op area, there's a little bit of a void. And what we want to try to do is fill that void by bringing in toxicology, looking for toxicological liabilities in molecules that like groups like Stan and Kevin are developing. So maybe they can fail early and fail fast, and then maybe increase their success rates there. And then also, like I said, apply that towards tumor efficacy studies in human tissues on a chip, organoids and spheroids. So that's sort of an area that we're going to be exploring and trying to move into in the future. And then my last slide then was just to thank the folks that I work with. We have a staff of 4 staff scientists that do all of the heavy screening and lifting. We've got an informatics team of 3 that really do work. We've got a great microscopist, and we've been working with Kurt Sang on the data analysis, rigor and reproducibility, and actually he'll be participating in the rigor and reproducibility conference that's coming up very soon. And that's where I'll stop. And sorry, my slides.
No,
sorry,
wonderful.
My slides stopped.
And your last slide did show up, Cliff, so I think
Okay.
that was good. So we
Fantastic.
got to see the various people that you're working with, or that are
Great. Better late than never.
Yep. No, I appreciate it. So thanks. So we're going to now turn it over to Kevin Dalby. And Kevin, I'm going to try to get this right, but correct me if I'm wrong. If I understand, you are currently the Johnson & Johnson Centennial Professor of Chemical Biology and Medicinal Chemistry in the College of Pharmacy and the Department of Oncology at the Dell Medical School at the University of Texas in Austin. Is that close enough?
That's what it says.
And that is a mouthful. But in addition to that, Kevin also directs and here to discuss with us his CPRIT-funded core program focused on targeted therapeutics and drug discovery. And this is based in UT Austin, and we're delighted that he can join us, even if it's just virtually. So welcome, Kevin.
Thank you. Can you see the screen?
We can.
Well, thank you for the invitation to talk about what we're doing over in Austin. I am going to kind of take a slightly different slant from the previous talks, partly because I felt that I would probably just be duplicating some of the things that were presented. So what I should start out with saying is the work that Stan and Matt presented, they showed so much work, and they just said it was, they didn't really tell us how difficult it was. And I think one of the really important things to appreciate is this stuff is not easy. And so having a network of core facilities around the state is, I think, a huge benefit to so many workers, researchers out there in the state, not only for the ability to, use the machinery, but actually the expertise that's out there in all of these different cores. So I would actually encourage folk to look into all of the different cores that are out there, not just the ones that we're talking today. So here in Austin, we've got a program that we call the Targeted Therapeutic Drug Discovery and Development Program, which is even more of a mouthful than my title. This initiated maybe about 2008 in UT Austin, where a high throughput screening facility was sort of envisaged as something that was necessary. We didn't have one on campus, and so this program evolved from having the facility back then. So we're funded through CPRIT, and there's myself and Unjong Cho, who does most of the work in the actual facility. She oversees the facility in the high throughput screening, and then we have a slew of other people in the core. So what do we bring to the community? So we're based on the campus in Austin, and we now, of course, have a medical school. It's probably been here about 5 years now, and so now we're starting to see a lot more interest in locally in projects, that are related to discovering drugs for cancer. And so one of the things that we can bring to folk are this access to the expertise in assay development. And as I said, this stuff is not easy, and the motivation for doing this was primarily to just imagine what it must be like to have the insight to identify a really potentially interesting drug candidate, and not knowing where to go after that. And one of the things that I've always reminded myself is that the expertise that I have is very different from the expertise that somebody else has. And I think really the motivations for these cores are to for us to kind of exchange the information that we have. So I was trained as a chemist, and one thing that I've really appreciated over time working with people is if you don't have a training in chemistry, then it's almost like a black box. And so we've worked hard to talk to people and explain to people what we're actually doing in terms of what these small molecules are, the properties of these small molecules, and how they interact with the potential targets that they're interested in. So the targets can be proteins, or they can be nucleic acids, and so what we can provide is a way to identify a way that a small molecule might interact with one of these molecules. And of course, not only that, it's not just an assay, but it's a high throughput assay, which involves quite a lot of work to get to be working in a really sort of efficient way, because we have maybe about 50,000 to 100,000 small molecules that we want to test. And I don't know if anybody's ever looked at an Excel sheet with 300 entries into a sheet, and suddenly you get down to the bottom and you scroll and it's already just too much. So imagine how to handle 50,000 entries or 50,000 small molecules. So the kind of expertise we bring is the management of these small molecules, and then the ability to test them all in these assays. And so these assays have to be developed in a very rigorous way to allow somebody to identify, to allow us to identify whether something is a hit or not. So we call a hit something that maybe inhibits an enzyme. And you only get one chance with these screens, because we screen so many molecules that we have to be able to identify the hits when they occur. And so So we're able to bring a real level of expertise to a person's project in exactly the same way that Matt and Stan do down in San Antonio. So we bring access.
Kevin?
Yeah.
Some of your verbal part is breaking up just a little bit, and it looks like your video is frozen. Could we?
No, I haven't moved the video, but the
No. Just not the video of your part. Could we? Do you mind if I turn off your video and see if that
sure.
improves the audio quality?
Yeah.
Sorry to interrupt you. I apologize.
No worries.
Back to you.
Can
Thank
you hear me now?
Yes, it does seem to be better.
Right. So the basic philosophy that I'm trying to get across is that we know that these things are difficult, and we want people to come to us to learn how to do these assays. And we appreciate that people come from many different backgrounds with wide varieties of expertise, and so we really do try to help take people to a new area altogether, you know, in doing these sort of screens. So so we bring assay development, then of course high throughput assays, the ability to do the high throughput assays, and one of the other things that we can bring to a project is computing, and I'm going to explain how we use that in a minute. And of course, we can also do some medicinal chemistry to support the project. So I had one slide which I kind of wanted to just present to talk through, because this kind of gives the overview of the program, really. So we have many molecules which are in our libraries, which we keep track of, and we have the ability to develop assays with individuals, and folk can come to us with an idea of an assay, or maybe they don't even know what the assay or the best assay would be. And so we work diligently to work out what we think are the best approaches to a problem. So our hope is that somebody will come to us with an interesting project, and we don't want somebody to not come to us for fear of not quite knowing how to move the project forward. And so our role, our responsibility, if you like, is to really enable researchers to get into this pipeline of drug discovery. So we're biochemists, we're chemists, and we can pretty much tackle any kind of assay out there. We have equipment to measure all sorts of spectroscopic changes, and we can monitor things in live cells as well. So we have this ability to develop the assays, and then, as I alluded to, what's really critical is to be able to turn an assay into this high throughput format where things need to be, pushed through at a pretty quick scale. So if we have maybe 50,000 molecules that we wanted to screen, the idea is that we would be able to develop a high throughput assay that would be able to get through those in at least a week. So the turnover of these assays is pretty fast. One of the other things that's really critical in drug discovery is what to do with the molecules that we identify as hits. And Matt alluded to that a little bit. And this is where a knowledge of the chemistry becomes important. But not only that, a knowledge of how small molecules interact with biopolymers like proteins and DNA. By understanding what to expect, we can focus in on the interesting molecules, and we can avoid mistakes. We can avoid follow-up on dead ends. So having that understanding of how small molecules interact with proteins, and really just having an innate understanding of biochemistry, I think really helps us take the screens and then actually turn them into really useful molecules for further development. Now, often when we find small molecules in these screens, one question is what to do with them next. And one of the things we can do is start thinking about making derivatives of them, which would be what Stan was discussing in terms of medicinal chemistry. But there are a number of other things one can do even before we get to that. So one thing could be just to look at the structures of the molecules that are in the screen, and look to see which molecules come through as the what we call the hit molecules, the molecules that are showing the most potency. And we can actually look at the structures and compare them, and we can identify similarities between those. And then beyond that, we can actually look for other compounds which are commercially available. So we can help people to go into databases of commercially available compounds and identify things that might work. And then that becomes a relatively cost-effective mechanism for finding other potential hits. And so by buying things that look similar and then testing them, we can get a pretty good structure-activity relationship out of the initial screens, and then this what I would call the chemoinformatics follow-up. And if we've got some structural information already on the target molecule, we can employ computational modeling. We have fantastic facilities here at UT, and a member of our program, Pengyu Ren, is a leader in computational modeling, and this allows us again to make decisions about what kind of molecules to make for lead development in the medicinal chemistry. So the medicinal chemistry is typically guided by structure, and we make analogs, and then we go back and we test them, and it's a sort of an iterative process. And so all of that requires a real understanding of how to do the assays and how to understand, how to kind of interpret the results. We have access to structural biology facilities at UT Austin too, so projects can go to structural biology. In fact, labs can do the structural biology themselves at the core facility here. So that allows us to sort of take the projects further. And we did have PK facilities, but we don't have those now. But instead, we have a CPRIT funded program over there in Houston now with the Dion Lang's group, which is it's CPRIT funded, and so we have a good connection there to send molecules for testing if a group is interested. So I won't. I don't want to go into all of the different projects and types of assays that we've been looking at. Our assays, the majority of them have been biochemical assays, enzymatic assays, looking at various different proteins involved in DNA replication and ubiquitination of proteins and so on and so forth, and protein kinases. I think that's really all I had to say. I just wanted to give you a sense of how we kind of can work with folk in a collaborative manner to sort of help get projects through this pipeline, and sort of give you a slightly different emphasis to things than the other presentations. I think, I was going to explain how understanding the mechanisms of a protein small molecule interaction can be important for designing novel new drugs, but I don't think I'll just stop there, actually.
Well, very good. Thank you very much, Kevin, for giving us an introduction and an overview of both the philosophy of your core and some of the resources. And so what I would like then, if Kevin and Matt and Stan and Cliff, if you guys could turn on your mics and turn on your cameras, and I may have, and if I I think you can all do that. I see Stan is on.
Yeah, you've shut me down.
I apologize. I am just not on top of my game. Let me start you up. See if that gets you back on.
Yeah.
And let's see, great. And who else? Matt, do we have you here? Yeah, I see Matt, and we have Cliff. Great. And for the audience then, or the participants, one of two ways: either unmute yourself if you can, unless I shut you down and you can't get back up, or throw your question in the chat. And these questions can either be for one of the cores, one of our speakers, to all of them to discuss your call. All right, and so we'll get a four-way discussion going and really pick the brains of these experts out there to help you out. And if you don't have any, I've taken a ton of notes because there are a lot of things that I'm curious about. So I'll turn it over to the audience first. And don't be shy. All right, well, I'm going to start then, and then if I don't see anyone, I may pick on someone. All right, from Marco Morris to everyone. GCMP facilities for clinical trials in Houston. So I guess the question is, do we have them? If not, where would you suggest? Can you guide people to the GMP facilities? And I'm guessing this is for small molecules, although Marco, if you want to clarify, are you thinking simply small molecules? Are you thinking biologics? Because two very different.
I mean, I think it for interest of everybody, both, I mean, molecules and biologics, yeah.
Okay, thank you. All right, gentlemen, thoughts on where to get this done?
I can start, I guess. So I don't know of any specific CGMP facilities in Houston. I've used people outside of Texas. And of course, we have a CGMP facility here in San Antonio at Southwest Research Institute that does both synthesis, CGMP synthesis and CGMP formulation work. And they do a very good job, and we've collaborated with them a lot.
Kevin? Thank you. Kevin, any thoughts on, have you?
The Southwest Institute was the only one that I knew of. There's nothing in Austin that I'm aware of right now.
Cliff, who would you point people to? Anyone in particular, or?
No, we haven't had to go that far, so we don't have any experience with it. So I can't point anybody at this point to that.
So, and I think Cambrix, if you're looking outside of Houston for national, they're not the cheapest, but they're usually pretty good. Biodura, I would say we've had pretty mixed results. So I think it's, before jumping in with one of these CGMP, definitely ask around. Other questions? Otherwise, I'd like the four of you to discuss. So if I'm a newbie or starting a new project, how do I get to your facilities? I mean, how do I get my project in front of you? How do I get your attention? How do you decide to help me out? And what's it going to cost me? So kind of big question, you know, kind of that big overview. How do I get use your resources, your expertise?
Well, at CIDD, originally we had this form that someone could, fill out online. And on that, there would be questions about, or at least, stating, what is the area of biology? What do you think the target is? Can you provide us with some papers so that we can, get better background? What is your funding situation? Those kinds of questions. We kind of, stopped doing that because we just started getting, people throwing a lot of spam at us, like filling out forms with just absolute garbage. So, I think, as of now, at least in the, in the San Antonio area, it's been a process, but, it's been developing. So, people, basically, at least for me, just contact me directly, and I set up a time to meet them, whether it's over the phone or through Zoom, now that, I'm pretty much remote now. There is really no cost for me to kind of spend time talking with them, reading papers, and really kind of getting an understanding of what their goals are for screening, either siRNA, CRISPR screening, or small molecules. So there's really no cost for that. But when it comes to getting an assay set up, there's other, kind of questions. So, does it require protein purification? So we now have protein purification capability. I found that, a lot of the proteins that you go and you purchase, they're really not of very high quality. So, we've, I have a pretty good person and fully set up for doing protein production, and we're kind of, augmenting that capability. So that ends up being a variable in terms of cost. And also Stan has, made probes available for enzymes that, promote hydrolysis of a probe. So, how you design that type of probe is something that a person could work with Stan on. And of course, there's a cost for that. But usually, for doing some initial assay development, trying to get some miniaturization, and even a preliminary screen of like 2,000 compounds, the cost for us is in the $3,000 to $4,000 area.
And
Which I think is, we probably aren't making money, there, because sometimes the really time-consuming part is this upfront work. But we get funding from CPRIT to provide this capability and these tools, and so in that regard, I think that we're pretty generous with our time and expertise in helping people.
And then just to follow along, and then I'll give everyone else a chance. So after the initial discussion, do you provide a timeline saying, all right, this is going to be the scope of the project, this is the timeline, and rough idea of the cost, kind of lay that out so people can decide?
Yeah. So, the timeline is also, it's going to be a function of how involved the screen is. That has to be obviously factored in as well. Initially up front, if we need to, put time into generating reagents and make sure that we have enough to follow through to the end of the screen, that is a time factor as well. But then, we've got a pretty good idea of obviously what our throughput's going to be. And we've got enough, equipment to really kind of move things along. And so, so that'll be a part of the equation. And then we have a kind of a pricing scheme where on a per compound basis, the more you screen, the less expensive it becomes.
Okay.
So.
No, very good. Kevin, how about you? How would people go about getting it on your radar? And then
so we do have a portal that is run by the Gulf Coast Consortium that was set up, I don't know, 6 or 7 years ago now, actually longer than that, which was the formal entry into the program. And people still do come in that way, but by and large, you know, we've got an internet, we've got websites, people find us, and then we get on the phone and we talk. And you know, I kind of sort of reiterate what Matt says. A lot of it is about talking, setting goals, and being on the same page. Because one of the things that we have to be able to do is tell somebody, actually, this is not good enough. It's not, we're not ready. We need to be comfortable to move through each stage of a process. So we have to have very honest conversations with people and set the expectations and be pretty clear up front where some cost elements may come in. You know, we tend to try and help younger investigators. We absorb some of the costs there. Through the funding, through the CPRIT funding, our labor costs are covered. So we don't charge for the labor. So really only what people would have to pay for would be materials like the plates. And I don't know what Matt's got, but I know Cliff and us, we have a liquid handler or a dispenser that doesn't use tips, that cuts down on the costs a lot as well. So we just work on trying to make something cost effective and set the goals. And Matt also talked about the proteins. By and large, we try to avoid commercial sources of the proteins and enzymes because that just gets us into a rabbit hole. So, you know, we stress up front quality reagents all round. Otherwise, you get really, you know, frustrated.
Okay. And so, Cliff, Stan, any additional thoughts on how we get into your expert facilities?
Ours is the same way. It all starts with a discussion. We get contacted by email, by telephone, word of mouth. The way into the core is through a meeting where we all get together. We try to understand the nature of the question that's being asked and why the screening might be an appropriate way to get at that. And then from there, if it is an avenue to explore, then we start to explore that avenue and determine what are realistic goals and endpoints for that. What can we actually do in our core? And we also evaluate whether we cannot do it in the core. And like I said, projects can, the nice thing about the GCC, and I was going to ask Matt and Stan the same thing, is projects can move seamlessly between institutions. So even though the GCC cores are all different institutions, there's no barriers between moving projects between them so that they can flow between them. Kevin and I have shared projects in the past where he's done some very fine development work, and then we were able to work with that investigator after they had worked with Kevin. And the other way, I mean, we're more than happy to there are projects that we can't. And that's why I like these forums, because I learned today more what Matt and Stan can do, and now what Kevin's doing as well. And when I have those opportunities to say that it's something I can't do, but I know someone who can, and then we can get in contact that way as well. But it takes a lot of discussion. You have to have realistic expectations. And I think that's the thing that we work really hard on with investigators, as you said, timelines and expectations as to how this is going to go. And to be honest, assay development, Kevin touched on it, is probably 90% of the project. Once you have a very good, robust, and reproducible assay, the screening goes fairly quick, but you spend a lot of time doing assay development. As far as cost goes, again, for cancer projects, our CPRIT funding covers almost all of the staff and equipment, and the only thing that we charge back is consumables and reagents, but that's at a cost it's going to cost anyway. So whether we provide it or you provide it, it doesn't make a difference to us, and it's going to be the cost of reagents and consumables anyway.
Any additional thoughts, Stan?
Yeah, so I'll add to that, I guess, a little bit. So first of all, it was a great point about the working across institutions, Clifford, because we don't have that problem either. It's been nice with the Health Science Center, with our collaborators. We share materials, and in fact, we developed an MOU with the Health Science Center and UTSA very early on because we knew IP was going to be an issue within the CIDD, and so that's actually pretty seamless. Going back to to how you get to us, I'll just add, it starts with a conversation, and I'll tell you, I spend a lot of my time working with people, not just about what their significant or specific needs might be, but what the big project goals are. I spend a lot of time analyzing chemical matter for people, and sort of looking and saying, okay, here's the possibilities with this lead or these structures. Here's the physical chemical property barriers you may want to consider. Those type of discussions then lead into, where we go with the project. And then for us, from a cost perspective, you can imagine a chemist on the chemistry side, we don't have a set cost because every synthesis is different, but we give custom costs for the work that we do. And again, we're supported by the CIPRIT as well, and so we give very, very reasonable costs for those, with cancer supported projects that are helped with the CIPRIT funding. But we give a custom estimate for every compound.
Very, very good. Now, we have a question in the chat. Emila, can you? I'll let you ask your question. If you can turn on your mic.
Yeah, thank you. First of all, yeah, I was just thanking Stan and Matt for another excellent presentation and great progress on their side. I was just curious about your ProTac platform, and you mentioned the design strategy based on the linker, but I was wondering a little bit more, if you're still working on it and expanding the size, and it's not an easy platform to set up, so just the challenges you face, and probably you already use it. I think that Matt alluded to that, so just wondering if there is any early success that you could share with us.
Yeah, hi Amelia, how are you?
I'm fine, hi.
Yeah, so I mean, this area as well as I do. So what our strategy here is following. There's probably 8 or 10 publications out there that highlight the importance that the linker plays in the properties of the molecule. Not only the activity for the particular target, but for bioavailability, permeability, solubility, and when you get into these ProTacs that are, very large in molecular weight. Building in those properties, really the only flexibility you have is in the linker, right? Because usually the ends are defined. So the approach we're taking is we've tried to develop some unique chemistries for making very rigid linkers that kind of sort of direct the two pieces in a specific vector or orientation, as well as making ones that are more flexible. And then within that, building in physical chemical properties that would improve solubility. So, keeping making the linkers not too lipophilic, increasing polar surface area, things of this nature. The other part of this is we're trying to make the platform very flexible from a synthetic point of view. So if you have a molecule that you're not sure where you can attach a ProTac to, but it's got a few functional groups, we're designing our chemistry such that the pieces of the linkers and the rigid templates are flexible with multiple functional groups. And so that's the focus. And we've made a library. I would say we have about 30 or 40 different linkers that we've made thus far. And we've used these for a couple of different programs. We've published a couple of papers on them, and I can send you those. We don't have any clinical success or preclinical success with these. Obviously, Avarnis Pharmaceuticals is one that's kind of at the front end of this right now. They're in phase 2 with the first ProTac in humans, and so it's going to be interesting to see what happens with these. But I would be glad to show you some of the chemistry we're doing and talk to you about it in specific if you're interested.
Wonderful. Thank you. Cliff, a quick question for you. Is there another question out there? Otherwise, I do have one. Well, if there is, just interrupt me. You won't hurt my feelings. But Cliff, can you comment on the zebrafish? All right, that's all. How many compounds do you typically think you'd be screening in those assays if one wanted to move into that platform?
The screens that we've done with that obviously have to be a collaboration with the investigator. The way it worked with the screening is the investigator would bring, these were 2-day embryo screens, so there's about 2 embryos per well in a 96 well plate, and we ended up screening in that particular project a little over 2,000 compounds over the course of about a month to do that. And we were screening probably once a week on that one. So it took about 4 weeks for us, 4 times to get through about 2,000 compounds. The limitation, it was all done by imaging. They had they were looking at vasculogenesis and the effect of environmental toxicants on vasculogenesis. And so they had a fluorescently tagged endo. So we were able to see with imaging that. We were also able to do a tox screen along with that because we could see the viability of the embryos. So it was a multiplexed endpoint, all done by imaging. Like I said, working with the investigator, they handled all of the embryos, getting them into the plates. We handled drug transfers, imaging, and data analysis for them. So it was a real nice collaboration between the 2 of us.
No, appreciate it.
Were those embryos in Houston? How did you arrange to get them into the lab?
We had an AUP with the university, but the embryos were brought to the lab. They stayed for 2 days in an incubator that the investigator provided, and then at the end of the conclusion, they euthanized and took away those, so we didn't have to deal with any of those issues. But A&M allowed us to do the screening itself.
Right, I see.
I'll have a pregnant pause here in case someone wants to jump in from the audience. Otherwise, I still have a question. This is the pregnant pause. Right. So out of curiosity and without, disclosing all your great ideas, the cores are working well. You gentlemen are collaborating and discussing things. What are we missing? All right. So I have 10 million bucks, that I'm willing to spend on a new core. So what do we need to further the, shall we say, the drug discovery and or development? And I realize that those are 2 very different things, but if I'm representing, I don't know, let's say the Parker Institute, and I want to set up another core in Texas, what would each of you recommend? 10 million bucks, 5 years. Thoughts on that? What do we need?
I would. So we've been thinking of ProTech as well, and there's a few groups around the country have been, developing cores. I think there's a group at Harvard I know quite well, and I think that it's such a big black box. Stan talked about the properties of the linker, but there are just so many aspects of what that linker does. There's a couple of incidents where the linker actually interacts with the molecules, and it's even allosteric. I think that we probably need a big push in computational modeling of these ternary complexes to really understand how these things come together, and that's going to need some big bucks and some big computing to make an impact. I don't know if it could make an impact, but to the extent that ProTax might work, and obviously we've only got 1 clinical trial right now. It's a potential because it could just open up so much if we just get over these first hurdles, especially in the areas of like degrading, neurodegenerative proteins and things like that. I mean, I don't think it's a big area for cancer particularly, because I think it's pretty easy to develop resistance in such a complex process. But neurodegeneration, I think, that's a big area. I think that could be huge.
Cliff, thoughts?
Yeah, our thoughts have been leaning more towards the pharmacology aspect of things, and it's like looking at that figure early on, you can see that we have a lot of early drug discovery work. There's a gap, we think, between those and getting them towards in vivo studies. As you're developing these ProTacs and other small molecules, going in vivo is an expensive proposition, and trying to take the best lead series forward or something like that is always a good question, is what are you going to do? So our thoughts have been leaning towards trying to develop the models that might allow an investigator who's developing drugs to do some quick evaluations for on and off target toxicology, and also in the case of cancer, because this would be CPRIT directed, tumor efficacy, is using human cells, using human tissues, where you're going to need to do that. So I guess looking for the liabilities that cause molecules to fail early in drug trials or preclinical tox.
I had a thought on that, Cliff, because I know you were thinking about that. And, one of the things that hasn't been addressed is the toxicity of combinations.
Yes.
And obviously in the cancer field, every drug is used in combination. And so,
Yes.
There doesn't seem to be a very good way to get from testing things in organoids to finding combinations that work to then, doing the toxicity studies.
Mm-hmm.
So I think we need new technology to do it.
Yeah. So those were our thoughts as well. Especially since we spend a lot of time doing combination studies. We're finding things, and some things have moved forward, but there's still the tox issues and the liabilities associated with that.
Getting back to the PROTECs, I mean, it's kind of such a rich area, and people are really only utilizing a couple E3 ligases. So one of the things that we're going to initiate is we've got 60 to 70 cancer cell lines. We've identified subset of E3 ligases, for siRNA screening. And so basically we're just going to use a cell viability assay to find out, okay, what are the E3 ligases that are required for cell viability in these different cancer cell lines, with the idea that if one is required for viability, then that would suggest that it's active and ubiquitinating proteins. And so that could then be a target for developing a new E3 targeting small molecule. That's, expanding the chemistry of, just the E3 targeting ligases, increasing that chemistry, I think, is also an area of development for the PROTECs. But, sometimes we find, we get good small molecules and we hand it off to people, and then, kind of just like, it doesn't move along as quickly as we would like. So, it's good to hear about the kind of mechanism of action capability, Cliff, that you were talking about. Maybe that's, a direction that I could kind of encourage people to move and contact you.
Right, definitely. Stan, there's another area that we just really didn't touch because we're all small molecule drug discovery people, and that's the biologics. Talking with Jiquan, and then, talking about what his interest has been with the design, with the engineered monoclonals, therapeutic monoclonals, is the scale up. We were having a talk about a week or so ago, and he was describing how expensive it is to scale those kinds of molecules to do in vivo studies or even beyond that. And so that might be another area that your money might be well invested in, as well is on the biologics side, which we didn't even discuss today.
No. Stan, any thoughts on how you're going to spend my 10 million?
Yeah, I have no problem spending it.
I know that.
No, I do have a couple thoughts on this, and the resources. There's two things here: the resources that you want to have, that you think you should have for a core facility, and I'm thinking on the drug development side, not the drug discovery side. Is I agree completely. Safety is a huge issue. So when if you look at the stats that are within pharma, 4 out of 5 compounds will attrit to some idiosyncratic tox finding, right? I mean, it's that's why they call it the valley of death. So how do we get through that when we're only in these academic circles or maybe working with a partner? And one of the things that I think we ought to rethink is that pharma did very well, I think, is when people think of development for tox or IND enabling tox, they go all in usually and look at cost for, you know, cGMP synthesis, GLP studies. I don't think that's necessary. You know, consider it more like a screen. And if we had a facility that could do that, both in vitro and in vivo tox studies, say just a short, you know, dose escalation, clinical sign toleration, maybe some necropsy, having that in a non-GLP just as a read on your molecule would really give a lot of confidence going into a full-blown tox study. That's one side. The other piece I think we could do more is in vitro tox screens. And one of the things we've been working on in San Antonio, and we're hoping to get this going with a new pharmacology core we're trying to develop within the CIDD, is ion channel cardiovascular safety screening. And for those of you familiar with the SIPA FDA initiative, this is going to be a real thing, I think, you know, if it continues to plow forward. So if you're developing a drug, it's no longer just hERG that you have to worry about. It's actually 6 or 7 ion channels. And so having that in vitro ion channel screen in an automated patch clamp capacity, I think would be a really good value to have. And we have instrumentation like that in San Antonio, and we're trying to develop that. So that would be the other piece that we're missing, I think we need to add in.
So for something like that, Stan, you're talking about, you know, significantly down the pipeline. Who do you think the clients would be? Would that be like 50-50 academia, industry, or how would you see that?
I would probably see it as half and half. You're right. I mean, you look at a lot of people in academics get held up at that point, right? And so that would be certainly a support mechanism. But if you made it cost efficient enough, like the high throughput ion channel screening, if we made that cost efficient enough, you know, you and I could use it for our medchem programs. You could have that as another data point early on.
Yeah.
I mean, you'd have to really get it nicely down because, I mean, Eurofins does this, you know, a few $1,000.
Yeah.
I mean, we can run those assays, a number of ion channels for a couple of thousand. So you'd have, if you to put it into a medium throughput, you definitely got to get the cost down or rethink how to do those. But
yeah.
All right. Well, very good.
I have a question. Can I ask a question still?
Please.
About toxicology and you know, de-risking compound early enough in the drug development pipeline. What about the use of human stem cells, the right platform? This is something that has attracted a lot of interest recently. So what do you guys think? And do you offer platform for that?
I wish we did. We actually put a proposal together on that. Dr. Jenny Shea is now down at UTSA. She used to be at UT Southwestern, and she's developed an entire stem cell platform and development of cardiomyocytes, et cetera. But our proposal was not funded, so we're gonna have to figure out another direction to go with that. I think it's a very viable approach.
Yeah, a lot of the models that we've been considering can also work with human iPSCs as well.
Yeah.
And Cliff, in your flow that you're developing, would this be something you would consider putting in, kind of isolating? I mean, it'd take a lot of cells passing through, but isolating some particular stem cells that could be then used for screening assays. Is this viable in your flow system?
The one that we have isn't a sorter. It's an analytical.
Oh, okay.
Well, we do have sorting capabilities, so we can do it. So we could isolate cells and then actually do the testing subsequent to that as well. So it could be a combined use. We have a flow core at the IBT that does flow sorting. So if you had a way to sort the cells, biomarker labeling, whatever you can use, then we could take those cells and immediately go to screening, which we could then also do by flow if need be. So if you had a co-culture or a complex model system, we could do that by flow. If it's an image-based system, we could move it into imaging. So it depends on what the endpoint would be.
Fernando, what do you think? Is that?
Yeah, I've heard recently a seminar about this ambitious plan of the EPA. I'm sure you know about it, to reduce of 20 to 30% the use of animal for tox studies by 2025 and to completely eliminate the use of animal for tox studies by 2035.
Yeah.
And so I don't know if you guys are aware of that, and I was kind of shocked. And so I was just, yeah, I just wanted to bring it to your attention. That's why I actually asked a question about the human-derived stem cell platform.
It's kind of interesting though, isn't it? Because, you know, the drugs don't fail at the animal tox stage all the time. They fail later. And so it's all about correlations. So, I mean, nobody knows the answer as to what's going to be most effective. And if we end up with a system where we use in vitro, you know, predictive models, and then something goes drastically wrong, we'll be back to animal studies.
Yeah, I mean, I agree. I thought that in any case, it's quite a drastic proposal.
Yeah, that's the 3R initiative that came out of OEDC a couple of years ago, and they've been working towards that. It's already been in cosmetic industry, has already been implemented quite a lot. It's sort of the idea behind what we were thinking as well, is if you could do a lot of in vitro work prior to going to animals, it does help reduce possibly the number of animals, because instead of taking maybe 2 lead series of molecules through, you take 1 because it looks so much more promising.
But the key is to have correlative data that is robust.
Right.
Right?
I'm talking with the FDA.
That's where the research needs to be done, is in actually developing the correlative models.
Mm-hmm.
You know, that might be something you could consider as part of your proposal, is to try and develop some kind of tool to, you know, predict.
And another, sorry, I think we have to go probably, but just a quick last question. In terms of toxicology, any QSAR based prediction software or algorithm that your guys are offering or implementing?
I'll give you my opinion. So yes, there's software out there you can use for toxicology for QSAR type issues, and it's based on compounds that have been studied and correlated to functional groups. But I think it's I wouldn't throw anything out because of it, because you know that they're not super predictive. I think every molecule is unique. You could take, and I've seen this happen, you could take a functional group that's not tolerated in one molecule, causes tox, and put in another, and it's completely fine. And so you just have to be cautious how you use them. One of the things I think they are useful for is very early on in a hit triage process. If you get a lot of hits out of an HTS and you've got 10 chemical series, you know, then that's a place where you can go and use something like that to help narrow down the leads that you follow.
Yeah.
Well, with that, and definitely appreciate the questions. Emails, addresses are around. They're on the GCC. I thank our panelists, our speakers, for giving us some overviews on the cores. Gentlemen, we appreciate it. And so again, as we say, please stay safe, stay healthy, stay sane. We will get through this eventually, and we will meet up for real beers and drinks sooner rather than later, right? So with that, we will close today's session, and we thank everyone for joining us. So take care.